Hello, Assalamualaikum. My name is Shahrul Azwan bin Muhammad Sabri. My metric number is 193343. This video is for FHM3109 forest pathology. My topic today is virus as a plant pathogen. So viruses were first discovered after the development of a porcelain filter called the Chamberlain Pasteur filter, which could remove all bacteria visible in the microscope from any liquid sample. In 1886, Adolf Meyer demonstrated that the disease of a tobacco plant, tobacco mosaic disease, could be transferred from a diseased plant to a healthy one via liquid plant extracts. In 1892, Dmitry Ivanovsky showed that this disease could be transmitted in the way even after the Chamberlain Pasteur filter had removed all the viable bacteria from the extract. Still, it was many years before it was proven that these filterable infectious agents were not simply very small bacteria, but were a new type of tiny disease-causing particle, that is, virus. The most commonly used system of virus classification was developed by Nobel Prize-winning biologist David Baltimore in the early 1970s. In addition to the differences in morphology and genetics, the Baltimore classification scheme groups viruses according to how the mRNA is produced during the replicative cycle of the virus. Viruses can contain double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA with a positive polarity or negative polarity, diploids, that is, two copies sRNA and partial dsDNA genomes. Now, please stay tuned and watch videos by the Amoeba sisters that I got from YouTube. Thank you, Amoeba sisters. Their illustration will give you a better understanding on virus replication process.
That makes virus replication or reproduction unique is that they can't replicate without a host. And it's their reproducing that tends to be a problem for its host, whether the host is you, in the case of the common cold, or an insect, plant, this bacterium. Viruses tend to be very specific for the host that they're going to infect, but anything that is alive can be a host because cells have the machinery that viruses need in order to replicate. We're going to talk about two types of viral replication cycles. The first one is the lytic cycle. In the lytic cycle, the virus, remember very selective, attaches to a host cell. It often binds to a receptor that this cell has, which gives it access to attach. The virus can inject either its DNA or RNA, depending on what kind of genetic material it has, into the cell. Some types of viruses are actually taken inside the cell themselves. Now, you would think that the cell would notice viral DNA or RNA or an entire virus that has been taken in, but in many cases, it does not. It takes the genetic material from the virus and it starts following the instructions, which in this case is very bad because the instructions tell it to make copies of the virus. The cell uses its own resources to start building. It starts making so many copies of the virus that it can cause the cell membrane of the host to rupture, explode, lyse. So what happens is now that these new viral copies, they can get out of the cell and go and infect other cells. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for the cell. The cell cannot survive without its cell membrane. The other cycle is called the lysogenic cycle, and I like to remember this is the longer word, so I like to think of this as the longer, sneaky cycle. Viruses that go through this cycle tend to do the same thing at the beginning. They inject their genetic material, but this time the genetic material stays hidden in the host genetic material. What happens is that when the host makes new cells, it replicates its own genetic material and the viral genetic material. Then those daughter cells make new cells, and they also replicate their own genetic material and the viral genetic material, and so on. This is the lysogenic cycle. That may not seem like a problem, but it really becomes one if the virus gets triggered to go into the lytic cycle from that point, because then all those cells that had the viral genetic material can start assembling viruses. And as far as what triggers it to go into the lytic cycle, it can vary. It could be a chemical trigger, for example, or a lack of food for the host.
Virus transmission. Some important animals and human viruses can be spread through aerosols. The virus have a machinery to enter the animal cells directly by fusing with the cell membrane. By contrast, plant cells have a robust cell wall and viruses cannot penetrate them unaided. Most plant viruses are therefore transmitted by the vector organism that feeds on the plant or are introduced through wounds made For example, during cultural operations like pruning, a small number of viruses can be transmitted through pollen to the seeds, like barley stripe mosaic virus, while many that cause systematic infection accumulate in vegetatively propagated crops. The major vectors of plant viruses are insects, nematodes, plasmodiums, and mites. Viruses have an intimate relationship with their host. They are completely dependent on cells for every phase of their life cycle. Most viruses are probably commensal, meaning that they obtain what they need from the host without causing any harm. Some viruses have a mutualist relationship with their host, providing benefit their host cannot live without while also benefiting from the host. Some viruses are true mutualists, meaning that they are provide a benefit to their host. This latter might be common, but there are yet only a few well-studied examples. Plants have a very different system of immunity. Innate response to viruses are sometimes specific for the virus and the plant host. For example, some viruses trigger a response in plant that keeps the virus in the initially infected cells and prevents it from moving to other tissue. These are called local lesion responses, and sometimes they produce yellow spots around the initial site of infection, or sometimes the cell around the infecting viruses are killed, leaving spots of dead tissue. Some viruses trigger an innate response that also affects other pathogens, so the plants become primed to defend against other invaders. One example of the plant viruses is tobacco mosaic virus. It is different from coronavirus because the tobacco mosaic virus is rod-shaped and made up only of genes and protein. Others, like the COVID-19 coronavirus, are round and wrapped in a fatty envelope that makes them especially susceptible to soap when you wash your hands. Well, that's all from me. Thank you and hope you enjoy this video. Bye bye.